Hello, I'm Bretton, one of our MCAT tutors here at Inspira Advantage, where we help students get accepted into medical school and other professional programs. And welcome back to another episode of MCAT Bites. Today, we're going to explore the transmission of genetic information from the gene to the protein. This process is fundamental to understanding how genes dictate cellular functions and ultimately impact an organism's phenotype. By the end of this video, you'll have a solid grasp of transcription, translation, and the regulation of gene expression. So let's get started. Beginning with DNA, or deoxyribonucleic acid. It contains the genetic instructions for the development, functioning, growth, and reproduction of all known organisms. It is composed of two strands forming a double helix, with each strand consisting of nucleotides. Nucleotides are shown here in yellow, blue, orange, and red. Each nucleotide contains a phosphate group in red, a deoxyribose sugar in blue, and the nitrogenous base, the A, G, T, and C. Altogether, these three things make up a nucleotide. So one nucleotide, two nucleotide, 3, 4, 5, 6, so on and so forth. And you'll notice that they bind A to T and G to C, and this is true forever. So let's talk about transcription. This is going from DNA to mRNA, where the M stands for messenger, so messenger RNA. Transcription is the process of copying a gene's DNA sequence into messenger RNA. And it's not quite copying because we're going to switch all the T's for R. This process is occurring in the nucleus of eukaryotic cells, and the steps involved in transcription are, first, we've got initiation. This is when RNA polymerase is going to bind to the promoter region of the gene, causing the gene strand to unwind. We've got elongation, which is where RNA polymerase will bind to the template strand of DNA and synthesize a complementary strand by adding RNA nucleotides. And then it's got to end, right? So termination. That's when the RNA polymerase is going to reach a terminator sequence, which will cause it to be released. And once RNA polymerase is gone, the newly synthesized mRNA will detach from the DNA, where it will ultimately be translated. But before mRNA can be translated into a protein in eukaryotes, it's going to undergo processing. A couple things it needs to do. First, we've got 5' capping. So here's our portion of the 5', and we'll see there's a little pink cap on it. So that cap is made up of GTP. It's a modified guanine nucleotide. Basically what it's doing is protecting it from degradation and aiding in the ribosome identifying and binding to it. Then we've got polyadenylation. Now this is just a fancy way to say sticking on a bunch of adenosines. Poly means a lot, and adenylation, adding adenosines. So once you know what it means, it makes sense. You just slap a ton of A's on the 3' end. And after we've stuck on our 5' caps and polyadenylated the mRNA, then we can get into splicing. So during splicing, we're going to remove the introns, which are non-coding regions. After the introns are removed, we're going to stick together the exons, the coding regions. That's shown in green in this diagram. So the introns are orange, we're going to get rid of them. We just want to keep the exons. And once we have our completed strand, now we can translate. Translation is the process by which mRNA sequence is used to synthesize a protein, or a peptide. This occurs in the cytoplasm on ribosomes. The steps involved in translation are, first, initiation again. So the small ribosomal subunit will bind mRNA near the start codon, which is AUG, and the initiator tRNA carrying methionine will bind to the start codon. So we always start with methionine. Then we begin elongating. The large ribosomal subunit will join, and amino acids are added one by one to the growing polypeptide chain. Each tRNA brings a specific amino acid to the ribosome, matching its anticodon with the mRNA codon. And once that's done, we're going to terminate, and this happens when we hit a stop codon. The 3 stop codons you need to know for the MCAT are UAA, UAG, and UGA. This is something I would recommend memorizing. Once the stop codons hit, the ribosome releases the completed polypeptide chain, and the translation machinery will disassemble. So how do we regulate all this? Well, gene expression is tightly regulated to ensure that proteins are produced at the right time and in the right amounts. Regulation can occur in various stages. The first is at the level of transcription, or transcriptional regulation. Transcription factors and enhancers/repressors will influence the initiation of transcription. For example, the lac operon in E. coli regulates lactose metabolism genes in response to lactose availability. We could also regulate after transcription, known as a post-transcription regulator. This involves things like alternative splicing, mRNA stability, and RNA interference, which can relegate mRNA availability. So in other words, if we mess with the 5' cap, or more often poly A tails, that can really mess with stability of RNA. And if the RNA isn't stable as long, well, we can't make as much protein from it. We've got translational regulation, which are factors that influence the initiation of translation and mRNA availability to affect protein synthesis. And finally, post-translational regulations introduce protein modifications, folding, and degradation will determine the final functional form of the protein. Let's apply what we learned with an example problem. I want you to describe the process by which a gene in DNA is transcribed and translated into protein. You can use this diagram to help you. Tell it to a friend, yourself, or just think about it. I'll tell you the answer in 3, 2, 1. The gene is first transcribed into mRNA by RNA polymerase in the nucleus. The mRNA will then undergo processing, 5' capping, polyadenylation, splicing, and is then transported to the cytoplasm. In the cytoplasm, the mRNA is translated by ribosomes into a protein with tRNA molecules bringing amino acids to form a polypeptide chain. Now I want you to explain the importance of mRNA processing in eukaryotic cells. I'll give you the answer in 3, 2, 1. mRNA processing increases the stability and translation efficiency of mRNA. The 5' cap protects the mRNA from degradation and aids in ribosome binding. The poly A tail enhances stability and facilitates export from the nucleus. Splicing removes introns, things we don't want to code, into proteins, allowing the mRNA to encode the correct protein. Understanding the transmission of genetic information from the gene to the protein is essential for mastering molecular biology and genetics. The process of transcription, mRNA processing, translation, and gene expression regulation are fundamental to cellular function and organismal development. Even more importantly, this can help you get a ton of more questions correct on the biochem sections of the MCAT. So keep studying these MCAT concepts and you'll be well prepared for your exam and your future medical career. Thank you so much for watching, and I'll see you next time.